Hello everyone, and welcome to the podcast. Learn about WoundSens project through its researchers. In this episode, EvoEnzyme scientists are going to share the main role we are developing in the project. WoundSens' mission is to overcome current limitations in the field of smart wound dressings through an innovative approach. Thus, EvoEnzyme brings to WoundSens its expertise in enzyme engineering and biocatalysis, leading the development of enzymes that will act as biosensors on the wound's microenvironment. Along these lines, we are heading the work package 3, which is actually involving the development of a directed evolution platform for oxidoreductase enzymes. One of my teammates will continue to tell us more about this and about his role as a researcher within the project.
Sure, so my role is to investigate and develop a kind of enzyme with optimal performance on the electrospun fiber materials. It must be able to generate a sensitive and robust signal for continuous wound care monitoring. More specifically, we start by choosing the organisms that contain the type of enzyme of interest. In this case, we select the enzymes of certain fungi and bacteria based on parameters of specific activity and thermostability. The next steps are to find a host that properly expresses the enzymes. We usually test bacteria, such as Escherichia coli, or yeast, followed by a method that allows us to purify the enzymes. Once here, we perform a biochemical characterization of the enzymes in terms of enzyme activity, selectivity or affinity for the substrate, thermostability or pH profile, among others, to identify the best candidate that meets the requirements. For this, we also test and validate different screening methods which are used to perform directed evolution campaigns aimed to achieve variants of the proposed enzymes. Thus, we are trying to obtain an enzyme that is functionally resistant to the microenvironment of the bandage and the wound, for which we have to consider all the media in which the enzyme will be present. In this way, we are testing the effect of different types of solvents, such as those used for electrospinning, as well as other chemicals on enzyme activity and stability.
Yes, it really is a demanding and laborious work. So what inspires you as a researcher about WoundSens?
Among the main aspects that most motivate me is that it's a truly innovative initiative, which is something no one has done before since it combines science from 3 different fields to achieve an unique goal. This is incredibly fulfilling for me because I get to learn a lot from researchers from diverse backgrounds, experiences, and perspectives, for example, on the electrospinning side. I think that's precisely what makes it both challenging and enriching. I believe the most inspiring thing about working on WoundSens is the thought that this solution will ultimately help patients achieve a better quality of life and well-being. And you, what do you hope to achieve through the WoundSens project?
I couldn't agree more with you. I trust that every result we are achieving right now will lead to a paradigm shift in wound dressing. WoundSens will contribute significantly to the progress in healthcare, improving the quality of life for those suffering from chronic wounds. Also, working on this project makes us even more aware of the versatility and immense potential of enzyme biotechnology. To all our listeners, thank you for joining us. To continue learning about this initiative, visit the official website and follow our journey on the project's official channels like LinkedIn.